Assalamualaikum. Hi, my name is Annur Sara Hisham S50327. I will introduce to you how I manage my project, which is a flora, species diversity and richness in residential area in my hometown, which is Bandar Kangkar, Pulai, Johor. So this project arrangement will be used the method classified as stratified random sampling using QUADRAT. I will be plotting two plots using a quadrat, and the first one will be in my yard. I will be plotting three subplot respectively, and then the second one will be outside of my house with a transect of 200 meters. First, we need 4 piles of wood and make it square to match a quadrat size, which 1 meter multiplied by 1 meter. Quadrat will be used as the division of plots in quadrat, and we will be count number of species found in those plots. And those are materials that I use for making a quadrat. After preparing the quadrat, bring the quadrat to the selected plot. Three subplots were randomly selected in my yard, and this is the first plot. Analyzed by naked eyes, there are 3 types of plant, and we will be identify the species right after sampling. This is the simplest and most used sampling device. It's a square frame of wood, and then organisms within the quadrat are counted. Thread used to be the subdivide the area within a quadrat. Count each species that grow in the plots of quadrat. Remember to take photos of quadrats placed on the plots and plucking the species found in those plots as a sample. Pluck the species outside of the quadrat for identification. There are 3 species found in plot 1 in my yard. Repeat the method to other plots, and thus the sample goes to paper bags for further identification. Take photos and also photos of sample of species collected in the plots. This method also used for outdoor areas to balance the study of plants distribution in these residential areas. Randomly place the quadrat on ground and remarks the species found. In plot 1, which randomly selected areas, thus far found 3 species. There goes second plot in outdoor areas, and also photos were taken exactly on the quadrat were placed. Count species found within plots and collect the samples from outside of the quadrat. Data collected will be defined in percentage of ground area within the quadrat. Sample collected from species within quadrat will be identified as species name by morphological of the flora and leaves, referring to sources as reference. Shannon Diversity Index will be used to explain the flora, diversity and richness in this residential area. With that, thank you.